Okay, we pick up with a discussion of the stomach, which is probably one of the most underappreciated organs in the digestive system because it allows us to do something that we take for granted every day, which is to feed in a discontinuous fashion, meaning that we can consume a meal, be finished eating the meal, and then carry out our activities of daily living. If we didn't have a stomach, we wouldn't be able to do that. We would have to almost continuously eat in order to get enough nutrition to keep the body going. So the stomach was a big leap forward in evolution. If you look at animals that don't have a stomach, their lives are spent essentially eating and producing waste. An example of such an organism would be an earthworm. So let's look at the stomach. It's got a couple of openings. The gastroesophageal sphincter opens into the esophagus. This is also sometimes called the cardiac sphincter because of its proximity to the heart. The pyloric sphincter empties into the duodenum. Parts include the cardia, the fundus, the body, and the pyloric area, which consists of the antrum and the pyloric canal. The greater and lesser curvatures are indicated here. Okay, see the lesser and the greater curvature. And these are attachment sites for membranes called omenta, which contain connective tissue and are imbued, especially in the case of the greater omentum, with a considerable amount of fat. There are sphincters that control the entrance and exit of food into and out of the stomach, the cardiac or lower esophageal sphincter, and the pyloric sphincter. Now, one of the things that we need to appreciate about the stomach is the fact that it produces very corrosive secretions called gastric juice, and the stomach is designed to handle those secretions like no other organ in the body. So as long as we confine the stomach secretions to the stomach itself, we're in generally good shape. The layers of the stomach are adapted to both produce and protect the lining from these secretions. The layers include the serosa, or visceral peritoneum, the muscularis, which is smooth muscle that consists of an oblique, a circular, and a longitudinal layer, and the submucosa, which is primarily lymphatic tissue, blood vessels, and nerves, and also there are glands in this region as well, and the mucosal layer, which is what's in contact with the gastric secretions, and also what's responsible for producing the bulk of the gastric secretions. If we look at the surface of the stomach, we also notice wrinkles called rugae, which are present when the stomach is relatively empty and disappear as the stomach begins to fill with food and liquid. If we look at the histology of the stomach in detail, what we see are little openings called gastric pits that are lined with simple columnar epithelia. The cells in the gastric pits produce mucus, hormones, and also pepsin, which is a powerful proteolytic enzyme, and hydrochloric acid. And all that together acts, number one, to facilitate chemical breakdown of the food. Number two, it acts as a sterilizing region for the food before it enters the duodenum. The mucus protects the stomach lining from the acid and the digestive enzymes so that the stomach doesn't erode its own lining as rapidly as it otherwise would. The mucous neck produces the mucus. The parietal cells make the hydrochloric acid and intrinsic factor, and the chief cells make the pepsinogen, which is the inactive form of pepsin. One of the things that we have to appreciate about a protease is that it's going to break down any protein it comes into contact with. So one of the recurring themes that we're going to see with proteolytic enzymes is that they're usually made as inactive zymogens, and they only become activated when they hit the lumen of the tract. At that point, they can begin to break down the protein content in the food that we eat. If they were activated sooner, they could damage the cell that actually secretes them. There's also regulatory hormones that are made by endocrine cells that line these gastric pits, Enterochromaffin cells produce histamine, which stimulates acid secretion, and the gastrin-containing cells generate gastrin. Somatostatin-containing cells make somatostatin, and this inhibits gastrin and insulin secretion. And you can see again what the stomach lining looks like. This is the cutaway diagram, and this is the actual histology of the lining. So what are some of the stomach secretions that help it do its job? Well, the chyme is what happens to the bolus after it's in contact with the gastric juice. It's basically the liquefied food. The mucus is made by the surface and the neck mucus cells. There's both a viscous and alkaline property to the mucus. It protects from the acidic chyme and pepsin so that the lining stays intact longer. Irritation of the mucosa causes more mucus to be produced. Intrinsic factor is made by parietal cells, and it binds with B12 and allows it to absorb into the blood so that it can be used for things like the manufacture of DNA, which is critical in the production of red blood cells. This is why in individuals that lack intrinsic factor, they often suffer from a condition known as pernicious anemia, where they simply have a low red count due to the fact that they can't generate erythrocytes. Hydrochloric acid is made by the parietal cells. It is a sterilant. It also stops carb digestion by inactivating salivary amylase that was produced in the oral cavity, and it denatures proteins and also helps to convert the inactive pepsinogen into the active protease pepsin. Pepsinogen is packaged into zymogen granules that's released in the process of exocytosis, and it catalyzes the breaking of covalent bonds in specific amino acid sequences in the proteins that are in the food that we eat. And so here's a look at how hydrochloric acid production is accomplished by the parietal cells in the gastric pits of the stomach lining.
Hydrochloric acid is produced in parietal cells through a complex series of reactions. Carbon dioxide diffuses into the parietal cell, and the enzyme carbonic anhydrase catalyzes a reaction between the carbon dioxide and water to form carbonic acid. Carbonic acid dissociates into bicarbonate ion and hydrogen ion, and the bicarbonate ion is transported back into the bloodstream. An ion exchange molecule in the plasma membrane exchanges bicarbonate going out for chloride coming in. The hydrogen ions are actively transported into the duct of the gastric gland, and the negatively charged chloride ions diffuse with the positively charged hydrogen ions. Potassium ions are counter-transported into the parietal cell in exchange for hydrogen ions. The net result is production of hydrochloric acid in the parietal cells and its secretion into the duct of the gastric gland.
And here's a look at the three phases of gastric secretion.
Regulation of stomach secretion is divided into three phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food sends nervous impulses to the medulla oblongata. These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsin in the stomach. The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach. This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsin secretion in the upper and middle parts of the stomach. In the gastric phase of gastric secretion, food has entered and distended the stomach. This distension activates a parasympathetic reflex via the medulla oblongata. and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsin. In the intestinal phase of gastric secretion, chyme has entered the duodenum, so gastric secretion is no longer needed. When the chyme contains lipids from the digestion of fats or contains enough hydrochloric acid to bring its pH below 2, gastric secretion is inhibited. The lipids and hydrogen ions inhibit gastric secretion by 3 simultaneous actions. First, they cause impulses to go to the medulla oblongata to decrease parasympathetic stimulation of the gastric glands. Secondly, they set up local reflexes via neurons in the wall of the gut that decrease gastric secretion. Thirdly, they cause the release of 3 local hormones which travel via the circulation to the gastric glands and inhibit their secretion.
And this is a chart showing you some of the functions of the gastrointestinal hormones. And again, I'm not going to go through everything on the chart. This is 24.3 in your text. Just make sure to know this material in preparation for upcoming PRS quizzes and for the exam. So let's take a look at some of the movements of the stomach. The stomach is a mixing chamber, and in order to adequately facilitate chemical and mechanical digestion, we're going to have to mix the bolus with the stomach secretions in order for, number one, the bolus to be broken up into smaller mechanical parts, okay, taking a big wet ball of food and making it into more of an emulsion. And the chemical digestion helps to begin to facilitate essentially the reduction of proteins into amino acids, of complex carbohydrates into simple sugars, and of fats into glycerol and fatty acids. The churning action along with the stomach secretions work together in order for this to be effective. This is a combination of mixing waves, which are 80% of the stomach movement, and peristaltic waves, which are pushing the chyme towards the pyloric sphincter, which is another 20% of the movement. Both the esophageal and the pyloric sphincters are normally closed when the stomach's doing this in order to confine the secretions to the stomach chamber. If these sphincters open prematurely, then the acidic chyme will regurgitate either into the esophagus in the case of an incompetent cardiac sphincter, or prematurely into the duodenum in the case of the pyloric sphincter. In either case, if this happens before that portion of the tract is ready for it, it's going to erode the lining of that part of the tract. For instance, if we have acid reflux into the esophagus, we have the potential to cause the esophageal lining to inflame and potentially bleed or even rupture, and is a dangerous situation characterized by the condition known as Barrett's esophagus. Scar tissue can be produced that closes the esophageal opening into the stomach off, and then it becomes difficult to get food into the stomach, and we often experience emesis. If we have premature emptying of chyme into the duodenum, we can have a duodenal ulcer, which can cause the rupture potentially of that part of the small intestine, letting food leak into the abdominal pelvic cavity, along again with that very corrosive gastric juice. So if we look at the phases, okay, we can see here that the mixing wave is initiated in the body of the stomach and it progresses towards the pyloric sphincter. The more fluid part of the chyme is pushed towards the pyloric sphincter, whereas the more solid center of the chyme squeezes past the peristaltic constriction and back towards the body of the stomach. Peristaltic waves move in the same direction and in the same way as mixing waves, but are stronger. Again, the more fluid part of the chyme is pushed towards the pyloric region, whereas the more solid center of the chyme squeezes past the peristaltic constriction and back towards the body of the stomach. The peristaltic contractions force a few millimeters of fluid chyme through the pyloric sphincter into the duodenum. Most of the chyme, including the more solid portion, is forced back towards the body of the stomach so that it can mix more effectively and eventually completely break down into liquid chyme. The timing of this squirting of chyme into the duodenum is critical. It has to be just slightly behind, a few milliseconds behind, the secretions that come from the gallbladder and from the pancreas in order to prep that region of the duodenum for the entry of the chyme. Among one of the most important secretions that come from the pancreas is bicarbonate, which helps to neutralize the stomach acid. We want that bicarbonate to sort of be in the duodenum just a brief period of time ahead of when the chyme empties, so that when the acid hits that region of the duodenum, it's immediately neutralized and doesn't damage the lining. In addition, the production of bile by the gallbladder, well, via the liver, the gallbladder is just a holding tank for the bile, helps to emulsify the fat that's contained in the chyme so that the fat digesting enzymes are more effective in doing their job. Those are the pancreatic lipases. The small intestine is the site of the greatest amount of digestion and absorption of nutrients and water. This is really what I would think of as the business end of the digestive system, because the whole point of digestion is actually accomplished in this region, and the purpose, of course, is to absorb the nutrients out of the macronutrients that we eat, along with things like vitamins and minerals, get them into the blood, and that blood, as we're going to find out a little later on, is going to be routed to the liver, where the liver will store the nutrient components as fat or as glycogen for later use, or in the event that the body needs those components for energy immediately, will pass them on to the rest of the body via the hepatic vein into the inferior vena cava. The different divisions include the duodenum, which is the first 10 inches beyond the pyloric sphincter, the jejunum, which is the next 2.5 meters, and the ileum, which is the next 3.5 meters. It's lined in the mucosal region with Peyer's patches or lymph nodules, due to the fact that we have to appreciate, even though the acid bath of the stomach is a sterilizing procedure, it doesn't kill necessarily all of the potentially pathogenic microbes in the food we eat. So we have to have lymphatic tissue in the mucosa all along the lining of the small intestine in the event that a pathogen were to penetrate. And it's in the ileum where we see the most pronounced concentrations of lymphatic tissue. These are the Peyer's patches. And if you were to ask, well, okay, what's in a Peyer's patch? Pretty straightforward. It's essentially a team-up of macrophages and lymphocytes designed, again, to alert the immune system to the presence of a pathogen. Remember, wherever we find lymphocytes, we're going to find macrophages reasonably nearby, because they work together to coordinate the immune response to the introduction of a pathogen. The duodenum curves to the left, runs across the head of the pancreas. In that curve, the major and minor duodenal papilla are openings to ducts from the liver and pancreas. And one of the things that we'll see when we take a peek at the duodenum and the accessory organs of digestion is that the fusion of the bile duct and the pancreatic duct into the duodenal papilla represents the potential, in the case of a blockage, to cause both organs to become inflamed if they're not able to send their secretions into the duodenum. And this can happen, for instance, as a result of gallstones. The other thing we have to understand about the small intestine is that this is where most of the nutrient absorption takes place. As a result, one of the things that's important is that we have to do a lot of secreting and also of absorbing. So we need a lot of surface area. So in order to increase the surface area, ridges called plicae circulares will dominate the small intestine, and on the plicae circulares will be finger-like projections called villi that are essentially embedded with lacteals and capillaries in order to absorb the nutrients that cross the lining of the small intestine. The lacteals are there to absorb primarily the fats, and the blood vessels are going to absorb the amino acids and the simple sugars. And you can see them in the diagram here. Okay, the green lacteals there for the absorption of fats, and the capillaries that surround them to pick up the amino acids and the sugars. The microvilli are the brush border that line the villi, and these are folds of the cell membranes of absorptive cells, and so what we see is a brush border on the surface of already a series of finger-like projections that dominates the inner lining of the intestine. So let's take a look at the mucosa and submucosa in the duodenum and talk about what they do. First of all, the mucosa is going to be that part of the duodenum that's in direct contact with the food and the secretions. Cells and glands that we find there are absorptive cells that have microvilli and make digestive enzymes in order to finish the process of chemically digesting the macronutrients, and they also absorb the digestive food. The goblet cells are the mucus makers, and the endocrine cells generate the regulatory hormones that stimulate the digestive tract to push the food through the pipe and also to trigger the secretion of the enzymes that are going to help to chemically break down the food. Granular cells help protect from bacteria, and there are also intestinal glands that we're going to find all along the lining of the GI that are essentially embedded in the mucosa at the bases of the villi. The duodenal glands are tubular mucus glands of the submucosa, and they open into the intestinal glands. And remember that the purpose of the mucus here is to lubricate the food and also, to some extent, protect the lining from the extensive mechanical abuse that it's going to take simply as a result of producing these powerful digestive enzymes, but also squeezing the food through the pipe. And this is one of the reasons why the mucosa has such a high mitotic index, because eventually it has to be replaced due to the physical abuse that it takes on a daily basis. The jejunum and the ileum are the next two segments of the small intestine. They have a gradual decrease in the diameter compared to the duodenum, as well as the thickness of the intestinal wall, the number of circular folds, and the number of villi the further away we get from the stomach. And the reason for this is that we've done most of the absorbing by the time we get really to the end of the jejunum, and so we don't need as much surface area as we did in the duodenum and the early parts of the jejunum. Because we've already transported the nutrients into the blood, the amount of surface area can then be reduced as we head towards the large intestine. It's the major site of nutrient absorption, and remember that the Peyer's patches are the lymphatic nodules that dominate the mucosa and the submucosa that are populated with lymphocytes and macrophages to guard against the introduction of pathogens into the blood. The ileocecal junction is simply where the ileum meets up with the large intestine, specifically where it meets up with the cecum. The ileocecal sphincter and ileocecal valve are what control the entry of waste products from the ileum into the cecum and subsequently into the colon. Secretions of the small intestine are primarily water-based, along with electrolytes and mucus. The mucus protects against digestive enzymes and stomach acid. The enzymes are bound to the membranes of the absorptive cells, and they help to break apart disaccharides, so they're called disaccharase. And monosaccharide products are what's going to be absorbed into the lining and will eventually find their way into the blood. The peptidases hydrolyze peptide bonds, while the nucleases break down RNA and DNA. Duodenal glands are generally stimulated by the vagus nerve, that's cranial nerve 10. Secretin also stimulates them, as does chemical or tactile irritation of the mucosa. Mixing and propulsion over short distances is critical in order to facilitate contact with the lining, but also to assist in the chemical breakdown of the food that we eat. Without sufficient mixing, this would be inhibited. Segmental contractions are going to help in the mixing process. If we look at the intestine, if we were to unroll it, what we would find is that there are periodic constrictions, and as the food shoots through these constrictions, it mixes more effectively with the secretions of the lining of the small intestine. Peristalsis helps to push the food, and remember that peristalsis is basically smooth muscle activity. And the ileosphinctal sphincter remains contracted until the peristaltic waves hit it, at which point it will relax and let the chyme move into the cecum, and this begins now the processing of fecal material. Cecal stretching causes a local reflex, and the ileocecal valve will constrict. This prevents more chyme from entering the cecum, and it increases digestion and absorption in the small intestine by slowing the progress of the chyme down. So it basically causes a temporary backup of the chyme into the small intestine in order to make certain that we've absorbed all the nutrient content. It also prevents backflow. The liver is probably one of the most important accessory organs of digestion in the entire digestive system, let alone your body. It's the largest internal organ. It has two large lobes, a left and a right, and a minor set of lobes, the caudate and the quadrate lobe. The porta hepatis is on the inferior surface, and it contains vessels, ducts, nerves that enter and exit the liver. If you look at the back of the liver, you can see that the hepatic artery and the hepatic portal vein enter here, and that the common hepatic duct exits here, and you can also find the gallbladder and the cystic duct nearby. So those are basically the three main leads that go in to the porta hepatis. The hepatic portal vein, artery, and nerve plexus also enter here. Lymphatic vessels and two hepatic ducts are going to exit. The ducts consist of the right and left hepatics, which unite to form the common hepatic, which empty into the cystic, which dumps directly into the gallbladder. And the purpose of the gallbladder is just to concentrate the bile so that it's a more effective emulsifying agent for the fat that comes into the duodenum from the stomach. Common bile duct is the union of the cystic duct and the common hepatic, and it's going to fuse with the pancreatic duct and empty directly into the duodenum via the greater duodenal papilla. And we can see the setup here. Note the hepatic ducts carry the bile that the liver makes. This combines to form the common hepatic duct, which is going to empty into the cystic duct, which empties into the gallbladder, where the bile becomes concentrated. The common bile duct and the pancreatic duct are going to combine to form the hepatopancreatic ampulla, which is controlled by the sphincter of Oddi. The hepatopancreatic ampulla empties the bile and pancreatic secretions into the duodenum at the duodenal papilla. You can see right here. The accessory pancreatic duct empties pancreatic secretions into the duodenum at the duodenal papilla. Okay, so you've got two openings for the pancreas to empty its contents into the duodenum. One of the things we have to appreciate about this is that, again, because of the way the plumbing is organized, if we block the area where the bile duct and the pancreatic duct fuse, when secretions from the stomach enter the duodenum and we trigger the pancreas and the gallbladder to empty their secretions into the duodenum, if they're blocked, we can inflame both organs, leading to pancreatitis and to inflammation of the gallbladder. Generally, we have to knock these gallstones free through lithotripsy or drugs that will help dissolve them, or in severe cases, we have to surgically resect. Liver lobules are very unusual looking. They're one of the few tissues in the body that has a geometric arrangement. They look like hexagons. Connective tissue septa branch from the portia into the interior, dividing it into lobules. Nerves, vessels, and ducts follow these septa. The lobules contain triads at the vertices of each part of the hexagon, which are made up of a bile canaliculus, a branch of the hepatic artery, and a branch of the hepatic portal vein. The central vein is in the center of the lobule and receives the blood that percolates through the sinusoids as the hepatocytes work on the blood content. The central veins unite to form the hepatic vein, which is the exit from the liver and empties into the inferior vena cava. Hepatic cords come out from the central vein and are made up of hepatocytes. The sinusoids are between the cords and are lined with endothelial cells and hepatic phagocytic cells, which help to digest cellular debris and pathogens. And the bile canaliculi are between the cells within the cords. The hepatocytes make bile, and they're there for storage of nutrients and for the conversion of one type of nutrient into another, and for the detoxification of ammonia, or nitrogen containing compounds that are the result of the breakdown of things like protein. By detoxification, things like the oxidation of alcohol, as well as the oxidative breakdown of recreational drugs, happens in the liver. In addition, deamination and transamination reactions happen in the liver, the purpose being to produce something that can be filtered by the kidneys and excreted from the body, so that we don't end up perturbing the body's pH as a result of the accumulation of these nitrogen-containing compounds. Phagocytosis also happens in the hepatocyte region. Again, this is to destroy worn-out cells and pathogens. We also make important blood components here. One of the most important is albumin, which helps the circulatory system, the blood in it, to retain its fluid volume because it's very osmotically active. If we lack albumin production, one of the things that will happen is it will upset the fluid balance between the tissues and the blood vessels, and fluid will tend to remain in the tissues, causing edema. An example of this is the condition known as ascites, in which, due to a lack of albumin in the blood, fluid weeps into the abdomen from the peritoneum that lines it, and the abdomen becomes highly distended. The liver is important in bile production, makes about 1 liter a day. This is composed of bile salts, which contain cholesterol, fats, and fat-soluble hormones, and lecithin. It neutralizes and dilutes the stomach acid, and it emulsifies fat. That's its probably most important role. Most are reabsorbed in the ileum. Secretin stimulates bile secretions, and it increases water and bicarb content in the bile. And this is important because you have to remember that the chyme that's coming from the stomach is extremely acidic. As a result, if we don't neutralize it as a result of contact with bicarbonate, we could erode a hole through the duodenal lining. Storage, very important role for the liver. It stores glycogen, fat, vitamins, copper, and iron, and hepatic portal blood comes to the liver from the small intestine, nutrient rich, right, imbued with things like amino acids and simple sugars. And then it's the job of the liver, of course, to convert those into a storage form for later use, essentially acting as your body's pantry. Nutrient interconversion is critical in the liver due to transamination and deamination reactions. We can take amino acids and we can transform them into Krebs cycle intermediates, and from there they can become part of the body's triglyceride content. Hydroxylation of vitamin D is an important compound that's made in the liver, which travels to the kidney where it's hydroxylated into its active form. And this is critical because vitamin D helps to mineralize bone. It's also a detoxifying reagent. Hepatocytes remove ammonia and convert it to urea, and they also oxidize ethanol, converting it eventually into acetyl-CoA, which enters the Krebs cycle. In addition, cytochrome P450 is an important detoxifying enzyme in the liver that helps to oxidize drugs and other toxins so that they're easily excreted by the kidneys. Phagocytosis happens in the liver where the Kupffer cells phagocytize worn out and dying red and white cells and bacteria. And it's also important in the manufacture of albumins, fibrinogen, globulins, heparin, and clotting factors. Now, just to review, we should touch on what each of these do because they're fairly important. If we look at each of these one by one, let's take here albumin first, right? So albumin is critical because it helps to maintain osmotic pressure in the blood. Fibrinogen, important as part of the clotting cascade. Globulins for defense. Heparin is an anticoagulant. And of course the clotting factors are also involved in clotting. Okay, so if we have liver defect, we have loss of liver function, all these processes will suffer. So let's take a look at blood and bile flow through the liver. Okay, the gallbladder is on the underside of the liver, and it's a place where bile is stored and concentrated. It's lined with mucosa that are folded into rugae. There's an inner muscularis and an outer serosal layer. The bile arrives from the liver and is stored there so that it can become concentrated. And CCK, that's produced by the intestine, along with vagal stimulation, causes the gallbladder to squeeze so that its contents, along with the pancreatic juice, can be introduced into the duodenum in order to intercept the chyme and, again, to facilitate chemical digestion of the macronutrients. Bile exits through the cystic duct and into the common bile duct. Sometimes the bile can sit too long in the gallbladder, and then it forms solid crystals known as gallstones. This can block the cystic duct and can also block the pancreatic duct, and it often occurs due to drastic dieting. This is just a look at how the nerve and the endocrine system control bile secretion and release. The vagus nerve is stimulated, causing the gallbladder to contract, and this releases bile into the duodenum along with pancreatic juice. Secretin, it's made by the duodenum and carried through the blood to the liver, stimulates bile secretion by the liver, which eventually goes to the gallbladder, and then from the gallbladder to the duodenum. And CCK, which is made by the duodenum and carried through the circulation to the gallbladder, stimulates it to squeeze, and it also makes the sphincters that lead out of the gallbladder relax. This results in the bile being released into the duodenum. Bile salts also stimulate bile secretion. Over 90% of bile salts are reabsorbed into the ileum and returned to the liver, and where they here in the liver will be reused. Okay, this will stimulate additional secretion of bile salts. Okay, that's all I've got for this block. Again, study this material in preparation for the PRS that you'll see shortly. See everybody later.